Okay, I have great pleasure in introducing Colin Ingram from the University of Newcastle in the UK to give the second plenary lecture of this afternoon. Colin is an experimental neuroscientist, and he's been involved in neuroinformatics activities at the local level. He's a very active member of our steering committee in the UK. And he's also chairing the most recent INCF program on metadata. And I suspect what he's going to be talking about today is his own community project that he's been developing, extremely successful, and I'll give the word to him now. So, Colin.
Okay, thanks very much, David. And thanks very much to the Programme Committee for inviting me to speak, and also a particular thanks to Shiro Zui and his local organisers for putting on such a fantastic meeting. So I've been given a really difficult job here. So firstly, I'm the person who is standing between you and the buses going off for the dinner. But also, I've got to follow that enormous tour de force that we've just heard from Marianne Martone. And that's a pretty difficult task. But she's really set the foundations for what I want to talk about today, and that is data sharing, but sharing in a practical way using neurophysiology. It's an area we haven't heard a lot about in this meeting. We've heard a lot about anatomical databases and so on. But there's a growing body of work trying to establish databases for sharing of neurophysiological data. And I'm going to tell you quite a lot about a project that I've been running for the last 4 years. Now, neurophysiologists actually make a lot of use of data sharing. They require work that shares data with analysts who can actually produce the tools that they use in the laboratory. They share data with theoreticians and modelers who model the data and then provide them with predictions about what their experimental data should show. So there's a good reason for needing an environment which allows people to share data. And current resources for neurophysiology are largely limited to software centres, analysis software centres, and to repositories of original file formats. But there have been relatively few attempts to produce a platform which actually allows people to share, manipulate, and analyse their data within one particular grid environment. And that doesn't just apply to neurophysiology, it also applies to other areas of neuroscience. At the moment, as I say, there are a number of attempts to produce databases that allow people to share data, and I'll just highlight a few of these. This is one that's been running for a number of years. It was set up by Dan Gardner. It's neurodatabase.org. What he did was he established a database. He wrote a very extensive metadata schema called BrainML, which allows people to describe the data, and that falls very much into what Marianne was just describing in terms of having a detailed information about the experiment. And he established a database that was searchable in order to find the particular files. He also addressed an issue which I'll come back to later on, which is that data in different file formats are difficult to exchange, and therefore he attempted to translate that into a common data format. The repository was mainly for published data, and therefore it had already been peer-reviewed and showed quality. However, if you go on to neurodatabase.org, you'll find that there's very limited data sets. There's only about 14 data sets that have been submitted to this database. And in fact, one of the issues that experimenters find is that despite having a very rich metadata, which we very much support, when it comes to submitting data into one of these repositories, if there's too high a barrier, then people are resistant to actually do so. The other database that I want to mention is the one that was set up by CRCNS, and Fritz Sommer is here at this meeting. This again is a database of original electrophysiological data. One of the issues at the moment, and I know Fritz is addressing this, is that it's not possible to actually view the data before downloading them. And he took completely the opposite view in setting up this database, that in fact there would be no structured metadata, but in fact there would be extensive free text documentation that would support all the files that were submitted, that would allow people to understand the data as deeply as possible. Most of the data has been published, but there are some original data in there which have not been published. Again, it's a relatively small set of data, but they are complete data sets and therefore very useful for modelers. And there are some data sets in there which are invaluable. For example, Buzaski's data on intracellular and extracellular recordings, which have been used for people as a benchmark for spike detection. I should also just point out that there are a couple of other efforts which are fairly new, and they're here at this meeting. So the work being done by the Keynote in developing up a data management and analysis platform is one of the posters here. And we heard earlier today in one of the spotlights the work that's being done at ATR for developing up an interface for data sharing. So that's really where we are at the moment in terms of sharing electrophysiological data. I haven't mentioned some of the projects which are sharing EEG data, which have some similar architecture, but this is largely experimental electrophysiological data. So the project that we've been running for the last 4 years is called CARMAN, stands for Code Analysis, Modelling and Repository for E-Neuroscience. And that is essentially what we aimed to do, is to produce a platform which would move beyond a database and create a functional portal, which our primary aim was to create this virtual laboratory which would allow people to share and analyse neurophysiological data. So as well as people submitting their data, there's the possibility to submit client-defined tools, so that people would produce services which would be uploaded, allowing extraction and analysis and modelling of the data. And as a consequence of actually having the data uploaded and analysed, we would, in a sense, as a byproduct, be producing a database or repository which would allow archiving, sharing and integration of the data. And what I want to do through this talk is actually show you some of the experiences we've had and highlight some of the issues that surround sharing of data. So the actual structure, the platform, is built on e-science initiatives that have been running in the UK for some time. So we already had, in terms of the compute architecture, a good basis for producing the compute cluster with workflow enactment engines and registry for storing the data and for storing metadata. And this was put together to produce our CANs. The one issue that we attempted to address to start off with is the one that Marianne was addressing, and that is the question of how do you describe the document. So we produced a discussion document which was circulated to a number of people. We had contributions and asked the question, what was the minimum information about a neuroscience investigation in electrophysiology that would allow people to understand what they were doing? And of course, the list was very extensive. However, we cut this down into eight domains, which we thought was the minimum. So the contact and context for the individual, what was the subject that we were looking at, or the study subject, where we were recording, what was the task, and we produced various terms under these 8 headings. Now, at the moment, the terms that are used, the lexicon that we use, is user-defined. So again, it's free text. But again, we should be working with INCF and with the NIF projects in order to make sure that this becomes aligned to the field. So in terms of data entry, using the compute architecture, we've been able to build a distributed and searchable database. It's built on SRB, the Storage Resource Broker, which allows federation of a number of servers. And at the moment we have 2, one based in York and one based in Newcastle, creating our own mini grid or cloud. The important thing about our data is that most of these data are unpublished, and therefore in this work environment where people are analysing, it's important that they have confidence that they have security over their data. And so access control ensures that there's only the individual involved or their partners are able to access the data. As I say, we have a minimal metadata schema. And I'm sorry, but most people will find this abhorrent, but we've made that actually optional implementation. Now, the reason for that is that produces a very low entry threshold. If you're collaborating with somebody and all you want to do is be able to upload your file in order to do the analysis, you don't want to spend several hours entering data, or even several minutes. So we've produced a very low entry threshold. Most people are entering the mini information, but importantly you can add to this afterwards with annotation, metadata editing, and you can add linked documents. So for example, when the paper comes out, you can link the publication to it. Now, I'm going to attempt today to do some live demonstrations actually on the portal running over in York. This is probably the thing you shouldn't do, but so here, this is the Carmen portal as it appears, and if you're entering data, that mini document has been turned into a template. As I say, the minimum thing you need to enter is simply a name. I'll call this file INCF, and a template is produced with a series of boxes. And even using a minimum information, you'll notice that there are 30 pages that the individual needs to go through. So we've made that easier. Once you've entered one sort of set of data as a template, you can call up the template and simply change the fields that need to be changed. So for example here, if I click on genetic characteristics, it tells me in the help here that I need to give things like polymorphisms, alleles and haplotypes in that particular box. So once you've entered all the information, you can upload the file, and one of the important things, as I said, is you can change the access rights. If I go to data here, the data fall into three categories. They're the public data, which people have said anybody can gain access to. There's your own personal files, so that's my username, NCDI, or there's shared files here, and these are people who have shared data with me. And if I were to look at any one of these, or look in public here, for example, I can find files, and simply by clicking on here, I can view the metadata associated with this. So this is now searching the database under electrophysiology, for example. I can look at the recording protocol, and I can find all the information that somebody's added about the amplifiers, filters, etc. So once that information is in there, I can gain access to it, and I can use the data. And if it's my file, as I said, the important thing is that you can actually control access. And that's very simple, because all I need to do, all I need to do is find a file, which is mine. So this one, and by clicking on this, it opens up a box. At the moment, everything is set as private. I can click public, in which case everybody can see it. Or if I click shared, I can choose who out of the 150 registered users here I want to share it with. So for example, if I wanted Raphael Ritz to have it, all I need to do is click there. And now Raphael can actually see the metadata associated with that particular file. Okay, so that's a relatively simple way to use the portal. Upload is very simple. So the other aspect of it is using analysis. This is key to our project. As I say, we didn't set out to just create a database, we wanted a work environment. And so within the community of people working on Carmen, we produced a number of analysis services which are wrapped as web services. The upload process, similar to data, captures the metadata about the service, and it automatically generates the input forms that are needed in order to run the service. And similarly, you can have access control. So if you've got a very novel algorithm and you don't want other people to use it just at the moment, you can actually keep it either private or just share it with a few individuals. Now, the advantage of working like this, to work on the web, means that in fact very little data needs to be moved around when you're collaborating with individuals. So we don't need to move large data files. For example, later on I'll talk about multi-electrode array data, which can get very large. If I'm collaborating, I don't want to keep sending the file to somebody. I can share the results of my analysis. People can see the results immediately they appear, and when I can conduct the analysis, I can do it in a very consistent manner, because the algorithm I use can be exactly the same as the other person, because you're doing it on the same server. And importantly, it exploits the available compute resource that we have on the server, and some of the things we'll do today will actually very much cut down the runtime. So when I run run a service. As I say, it's a very simple. Let me just go back to here. So running a service simply requires that I pick the service that I want to use, which under actions here, execute a service. In this case, what I want to do is create a movie, which we'll have a look at later on. I browse to find the file that I want to run. Sorry, the slow bit here is actually the connection between here and York. So I select the file. In this particular service, it's a filtering, so there's a filter file associated with this service as well. And I set the sampling frequency, which in fact is 1 kilohertz for this particular file. When it runs, I want to put the data into my own folder. And for the purposes of this, I'll give it a new folder, so I'll call it INCF, which is a very sensible thing to call it. Okay, so it's going to put the result in there, and it's going to run. And I can see that it's running, hopefully, he says, by just going into the service log, which now tells me that, in fact, here it is. It's queued and waiting to run. Okay, we'll come back to that later on. Okay, so we've been able to populate the platform with a number of basic analysis services such as filters, spike discriminators and detectors, cross-correlation, post-stimulus time interval histograms, etc. And these generate either graphical outputs, which we can download, or numerical outputs, which can go either into other analysis routines or simply be stored for later use. But one of the issues about this, and it became apparent straight away, is that there's a major problem with running services because of the variety of file formats, proprietary file formats that people use for recording. And currently, our services will only work with very specific input file formats that have been written for that particular service. And we have to rewrite the format for the service if it's going to use a different format. So to go around that, over the last 18 months or so, we've been working on an internal file format based on the NeuroShare DLL, which is called Neurophysiology Translational Format. It produces a MATLAB type file, a MAT type file, and this we can convert most of the proprietary formats into a single service, and this allows interoperability between services. It also means that we can export the files and other people can read them once they see our NDF format. Now, one of the key areas that we wanted to work on when setting up Carmen was not really single cell recording, but actually multi-electrode array data, because within the field of neurophysiology there's an increasing number of techniques which allow you to record greater numbers of neurons. This is fantastic because it allows you to analyze network properties of the brain and really gives a great insight into the temporal dynamics. So one of our aims has been to produce an infrastructure that supports the storage and analysis of MEA data. And what I'm going to do for most of the rest of the talk is talk about some data that we've been collecting through a retinal developmental recording consortium, which is headed up by Evelyn Cernagor, who's here at the meeting. She's based in Newcastle. But the data come from MEA recordings that have been made in mouse retina, and we heard yesterday about the fantastic anatomy of the retina. But the electrophysiology that comes out of that essentially two-dimensional structure of ganglion cells gives us enormous insight into what's happening during the plasticity period and in development. So working in groups from Seattle, Berkeley, Davis and Santa Cruz, which are all doing recordings, as well as the recordings which are made in Newcastle and Genoa, we've been building an infrastructure that allows us to share and analyse these data. So the first thing to say is once we've got data and we're sharing it, an important thing is to be able to visualize it. And so we've incorporated within Carmen, or associated with Carmen, is a thick client tool, a Signal Data Explorer, which actually allows you to look at the data. And at the moment, unfortunately, this is only a client tool, and therefore we can't actually run it on the web. That's our next process in the project. But we can download files and we can view them on our own PCs. So this is it takes a variety of different formats. It will take NDF, but it also takes multi-channel systems, for example. So here's a file. We'll just open this up. And this shows us a 64-channel multi-electrode array. And if we open this file, so this is now downloading it from York, and run this, we can see the activity on a number of electrodes. In fact, we only see a limited number here initially, but let's have a look. So here's channel 36. But the better thing to do is actually see this as a multi-electrode array. So this is a file of 64 channels of activity. Now, one of the key features of retinal activity during development are waves of spontaneous activity. And we just see here, let's go backwards, the onset of a wave of spontaneous activity. So most of the electrodes show silence up to this point. But here you can see propagating across the array these waves of activity, and we can see one channel down here. And I'll talk a lot about the way in which analyse these in a moment. So if we stop that just for a moment, here we've got one individual action potential. One of the good things about SDE is it includes a very powerful pattern matching tool, which allows us to search the data very rapidly. And here in this window, it's actually returned all the events which have this particular wave shape. So I can just click on this, and it will take me to the next event. And I can also do some simple things like looking at average waveforms simply by selecting the pattern to look at. So there's one there. Okay, so being able to see the files is very important. It allows people to understand whether or not they're data that they want to analyse. And as I say, at the moment they can only read certain file formats, but it's just being transitioned to full NDF compliance. But there are other visualization tools that we're running on the portal, including running movies. And similar to the analysis that I ran earlier, we can simply execute this. So we can run a movie to see how the activity looks. So simply look at this. So it asks for the file type. And here's the shared data from this collaboration. So the MEA data come from different laboratories. So here's Evelyn's data, Rachel Wong's data, Louis Chalupa's data, and we'll just have a look at one of these files. And as I say, one of the issues about interoperability is that, of course, at the moment we have to write this service for every different file type. This will disappear once NDF is deployed. So the data come from Newcastle, so we just run it on there, and let's just run. Okay, finish. And if we go into the service log, you can see now that, in fact, that laser service that we started earlier is completed. It's now running the So here it's completed the analysis of producing that movie file, and if I open the INCF folder where we're doing this, so MEA movie. Sorry, as I say, the slow bit is the link between here and so here. This is now generated at York. It's generated this movie of activity, and you can see occasional waves of activity that propagate across the electrode array. Most of the time, these are silent, and you're only seeing a small amount of activity. But then you see bursts like that, and then suddenly you'll see a wave here, for example, that's propagating over the array quite slowly. Okay. So that's fine. That was 64 channels, and in fact most of the data that we have is 64 channels, and multi-channel systems produce very nice arrays. But the challenge that we have is as fast as we're producing the infrastructures for this, the number of channels is going up. So Multi-Channel Systems now has a 256 channel array, and Keith Matheson, working with Alan Litka, who's also a part of this consortium, has been working with a 512 channel array. Now, Evelyn likes to set us some challenges, so most recently she's been collaborating with Luca Bertondini in Genoa, who has produced a CMOS MEA, which is now 64 by 64 channels, so this actually gives us 4,096 channel array. Now, the big advantage of this, of course, is that with a 21 micron electron spacing, 42 micron center to center, we're actually getting down to the point where we're almost at the level of individual neurons across the array, and the size of it allows us to do work across the entire, or what we call pan-retinal recording. So here's an image of a flattened mouse retina, this sort of white thing in the background, and superimposed on it is the shape of the array with the output from that particular analysis of activity. So I'm going to just get going with run that service. Sorry, it takes a second just to do. So I'm going to produce a movie now from the same thing, but again, this is now using the 4000 electrode array. Okay, and this time, of course, I'm running the same service as I just did, but of course this comes from IIT in Genoa, so I just have to change the file format. As I say, we're hoping to get around that in the future. So there it's running, and I'm just going to run that in the same folder. Come on, okay. Okay, so that's just running at the moment. So as well as being able to just look at the data, so we can look at patterns of activity, the key thing is to start doing detailed analysis. As I say, these waves of activity occur during the development of the retina, and we have services that allow the analysis of the patterns of activity and network properties to be made using the deployed services to calculate the parameters, and this allows us to compare between the groups. So again, we can run a simple service, and this service will actually do a number of things. It will produce what's called a four-plot, and the four-plot actually produces both an activity plot and also some data about interburst interval. So I'm going to do that on the one we saw, the pattern of activity. So here's, oops. Okay, so select that. MEA type, it's from Newcastle. This will produce both a CSV file, which you can then use for other analysis, or simply produces an output. We'll call that INCF. And run. Okay. So if I go into the service log now, oh, in fact, it's completed. Yep. So we've managed to complete all that. So here's our file. It's running very quickly. Okay, so this is the slow bit where it tries to download from York. So this actually shows us the pattern of activity analysed on that array, where we were looking at the waves. Sorry. Good. Okay, so what we see here, this is the 64 channels. Remember that the original one was done on 64 channels. Here's some bursts of activity averaged, and this is a raster plot showing the individual channels. And you can see here where there's alignment between the different channels, that's a wave of activity that's propagating across the electrode. So by doing this sort of analysis, we're able to make comparisons between data which would not otherwise be possible. If Rachel Wong did the analysis, even if we sent her the analysis routines and she ran them in her lab and then we ran them in ours, it would not necessarily be possible for us to compare those data. So by sharing the data on the web, we're able to compare directly. And here, for example, this is looking at cumulative burst duration for those activity that's happening at 5 days postnatal, and you can see something that has emerged out of this, and that is simply that burst duration is very much lab dependent, and so whilst there's a very tight distribution within the individual labs, which are color-coded here, they actually separate out. And so laboratories are getting different data, perhaps because of local environmental factors, and that doesn't apply here, for example, to burst interval. So as I say, analysing activity like this is important, but the high-density arrays provide us with a much greater detail than we get with 64 channels, where the electrode spacing is quite wide. So we can now actually just compare that, because we have available the data from the 64 channel array here, showing the bursts. We also have here analysis I actually run early because it takes a little while, and you can see the very fine detail that we get in terms of the propagation of these waves across the array as these spikes line up. We're also able to see the activity of these spikes, and there's the one that we saw earlier, that's the 64 channel, but now let's look at the quality of data that we actually get from using 4,000 channels. So this is now a recording that's made using the 4,000 array, and as you see, these little bursts of activity start to propagate, and you can get a lot of detail about exactly which cells are firing, how far they propagate across the retina. Here's one, a very nice distribution there, as these waves of activity. So this is the movie that we generated just a moment ago. I can tell you, because it's absolutely uniform when you run this. It takes 4 minutes and 45 seconds for this to run on our server, but if I ran this on my desktop, it takes just over an hour to generate some of these movies. Okay, so let's just go back to here. So we can get very fine detail of the activity of these propagations here, and we can look at the activity as they change across days. So each of these is a single retina using the high-density arrays, and you can see the propagation profiles change with age, becoming much faster as they get older. Of course, the problem we now have is that we're dealing with very high-dimensional space. So working with the group in Plymouth, the visualization lab, we're working to implement new tools that allow us to actually see the data in greater detail, including, for example, this fisheye lens, which looks at raster plots to actually blow them up. You can move this across. I won't show this live. And by comparing not only between groups, but between ages, we're able to get a much finer detail about the dynamics of these retinal waves. And here, as you see, here are the waves now in colour, seeing small waves that move much faster, but mostly we're on the periphery here at day 10, and they're very spatially restricted, whilst earlier in development, you get these much slower propagating patterns. And we can see that by doing centre of activity trajectories for network activity in the retina. And here are two examples on day 5, plotted with blue being the beginning of a trajectory and red being the end. you can see that they propagate slowly across a large part of the retina, whilst by day 10 they become very constrained to particular areas of the retina. And plotting that in terms of the number of clusters of these propagations and the number of waves per cluster, you can see there's a switch that occurs here between day 5 and day 10. And we're looking at what the underlying mechanisms are that cause that switch. So I've already shown you what we're trying to do in terms of coping with data that moves from perhaps 1 or 2 channels to 64 channels up to 4,000 channels. But when we're now reaching the point with these high density arrays that we're reaching the density of pixels that actually occur when you do ion and voltage sensitive dye recording. So we're also now starting to incorporate into Carmen the possibility of doing voltage sensitive dye recordings. And using our system, which has now a 100 by 100 pixel camera, we look at the 10,000 channels that we actually can generate. And let's have a look here at the movie that we actually generated at the beginning when I started the talk. So here's the laser service. So this laser service, what it does is it analyzes the activity within individual pixels. It first of all averages the first part of the recordings. It then subtracts that. It does a filtering backwards and forwards, and it then works out the gamma oscillations and then plots that here as a movie. You can see the individual. Oops. You can see the movie in a moment. But here it also generates individual pixel analysis. So this shows you an example of that individual pixel. Here's the raw recording. This is it after it's being filtered, and we are now getting gamma oscillations that we can see. And if we reduce that in a moment, we should be able to see what we've actually generated. Oops. He says, hopefully. Okay. Sorry, it's a very large file. Okay, so this is the movie that generated from 10,000 pixels, showing you the activity that's occurring across a piece of hippocampus, showing waves of propagation, but also within individual areas, the gamma oscillations that are occurring. And when we analyse, I won't do all the analysis now because it'll take a long time, but if we analyse those gamma oscillations and we look at the phase relationships within the dentate gyrus, for example, there's coincidence that occurs across the area here of the primary cells. So areas 1, 2 and 3 here are fairly coincident. But if we look at the phase shifts that occur between this area and further along the dentate gyrus, there's increasing phase differences, which shows that the slow propagation of activity of the waves of gamma oscillation that occur. This is in vitro, yeah. Okay, so this is in vitro slices from rat. And we're now doing the same analysis here of temporal cortical gamma oscillations, both in monkey and human cortical slices. The monkey and human slices are a very valuable resource to us in Newcastle, and not only do we do optical recordings, but we also record using 10 by 10 UTAR arrays, generating a large amount of activity, both in terms of single spikes, but we can also look at the rhythms that occur within different fields. So for example, in layer 5 we get oscillations, and in layer 2, and you can see the nested rhythms that actually occur at the different cortical layers. Now these data can all be generated online using Carmen, and as I say, the important thing here is valuable data that can then be shared with collaborators, and once published can be shared with the wider neuroscience community. We can also look at, by looking at correlations of gamma, within the cortex rather than in hippocampus, we can again look at long-range network connectivity that occurs within human cortex. Now, this sort of very high level of analysis gets more and more complex, and that's fine. The compute resource that's available to us makes that possible, but it's much easier, rather than running sequential analysis, to run this as a workflow. So, for example, here, going back to our 64-channel arrays in the retina, we can calculate spike times using spike sorters. We can produce a correlation matrix. We can calculate thresholds to look at the strongest connections. We can then, using those strongest connections, produce a network graph and calculate network measurements. So that's interesting in terms of looking at the network activity within the retina, but we really need to run that as a workflow. So once NDF has been deployed, we've also got a workflow enactment engine in Carmen, which involves a GUI interface that allows you to link together all the different services. And because of the interoperability, we can now run this analysis not as individual processes, but as a single workflow. We're currently improving the usability of Carmen. You might have thought that in fact our website looks a bit clunky, so on the basis of feedback that we've had from people, we're improving usability, so our portal will be updated fairly shortly. And this leads us to the question of why are we bothering to do this? What's actually the advantage to people? Well, I go back to the fact that, in fact, using our services deployed on the web, we can get consistent and reliable application of these. The use of the web actually leads us to the development of a repository, which is invaluable for these very high value and rare data, such as human cortical and macaque slices. I don't know what it's like here in Japan, but I'm sure in the US and certainly in the UK, our funding agencies are increasingly requiring us to make use of our research outputs, making them publicly available. But at the moment, there's no repository that allows you to do that other than the products like Carmen. So therefore, it not only produces a repository, it facilitates sharing and collaborative working, which can, if we are working across geographical distances, can actually be conducted in near real time. And the final point is that I had the word cloud in my title. It capitalises on the developments that are happening in cloud computing, particularly allowing access to large compute resource. So very fast analysis can be done, which would perhaps not be possible unless you have access to a large grid, for example, in your local university. So what about moving to the clouds? Well, moving to a cloud computing has great advantages as a business model. A concern for us all the time is the sustainability of these databases and these services. So cloud service providers like Microsoft and Amazon, Google, etc., they can reduce the infrastructure overhead and increase the chances of sustainability of resources like Carmen. And particularly the unlimited storage capacity, or essentially unlimited storage capacity of the cloud is a good advantage. But it would still require us to transfer the data across to the cloud, and it may be that a federated grid can reduce the need for uploading some of these large files. And it's possible that in the future a hybrid model may be adapted to allow us to actually go forward. So if anybody's interested in this project, and I hope there's a few electrophysiologists out there or people who are generating analysis tools, or theoreticians who would actually like to get access to some of these data, you can do so through the project website, www.carmen.org.uk. Simply click on the link here, which is beneath the picture of the development team at our last consortium meeting, and you can register. It's free for anybody to register as a user. So I'd just like to close by acknowledging the enormous amount of work that's been done in developing this over the last 4 years. I'm pleased to say we've got funding that will sustain the project for another 4 years. There's been tremendous work been done by the computing science teams at York and Newcastle, led by Jim Austin and Paul Watson. We have 150 users and project partners who are contributing data and analysis code to Carmen, and that's absolutely essential because without users, the project will die. And a particular thanks to the MEA consortium led by Evelyn Cernigor for their work in proving the utility of Carmen as a work tool and also producing some fantastic data. So thank you very much for listening.
Thank you. Well, thanks very much, Colin, for a really fascinating talk. We do have a few minutes for questions.
So I'm very curious about the workflow approach that you've begun to implement, and wondered, since it's very important, of course, to try and flag this data with an appropriate pedigree associated with, the metadata for everything that you've done, if you're thinking about, or if you have already implemented a provenance tracker, and if you're storing the workflows and making them available.
Yeah, we haven't implemented that at the moment, but part of the funding that we've just received for the next 4 years is to look at the sustainability of software and provenance for services, yes. Is
the workflow engine you're using home-built, or is this Taverna or Kepler or a commercial one?
It's none of those. It's been written in Newcastle. It's a new workflow.
Related to Mark's question and the analysis aspects within Carmen, is there a way for me to actually introspect the code that is running when I click on a certain service? Question number one. And then assuming I would like to repeat that analysis 2 years later on a different data set, but maybe the implementation of the analysis has changed, would I see that and could I roll back to the old one? And third question, could I take this as a starting point for my own development, and how would I be able to sneak in my code to run in this cloud?
So running your own code is not a problem. I mean, so you can do that now. You are registered. You can upload it. Simply go into. There's an upload for services. At the moment, there's a little bit of work that we have to do in wrapping it in York. It isn't entirely led by the code generator, but in the future, we're just writing a little GUI that allows that to happen automatically. So you can wrap your own code and run it directly. You mentioned provenance of software. Yeah. I mean, that's again part of what we're about to implement in the next phase of the project.
Yeah, and can I introspect the code? That was the first question. Can I see what's running?
Right, right. Let me see. The answer is you should be able to. In fact, you should be able to download the code. Something I don't do, and I'm afraid that's slightly beyond me.
I'm wondering what would be the workflow for a publication. So you do a lot of analysis on the very same data set, and then finally you have to produce some figures and then also statistics over all your analysis over many data sets. I don't see how you get this, so to say, out of the internet. And how do you play with your data?
Oh, okay. So. Well, that's an easy one. So if, for example, I hope it's an easy one. It was a danger, wasn't it, me demonstrating this thing live. So if I go into, it's in fact here. So here's a MAT file that's generated as part of this particular service. I can just click to download. Okay. Actually, I won't, because look how long it's going to take. So anyway, you can simply download the output from your file, from the analysis. So if you want to take it out of Carmen, so I'll cancel that because it'll take ages. So if you want to take say it's a CSV file and you want to run it then on your desktop, you can simply download it from Carmen. We don't currently envisage incorporating any statistics packages in here. I mean, it's not going to be a completely all singing and dancing web in that respect.
Okay, thanks.
Okay.
That was a lovely presentation. To me, this is a dramatic example of the transition from the static publication to a dynamic web-based world in which scientists will work. And it changes, I think, the way you would interact with the literature. You'd actually end up interacting with web services instead of the literature. So bravo. A question about turning this into a real-time service that doesn't require the user to register. It looks like, based upon that example we just saw there, you just need 40 times the horsepower to get it down from a long coffee break to a couple of eye blinks. How would you implement that so that you could do a real-time service?
Right, so we're looking at this at the moment because Mark Cunningham in Newcastle is collaborating with Mark Kramer over in Boston on some of the human gamma oscillation data. Because we get the data from neurosurgery and then have to run the experiment over that day, we need to understand some of the data before we decide what the next step is in terms of the experiment. So we're looking at packaging the data and uploading it onto Carmen. It can then be analysed. I mean, Mark Kramer in Boston can then actually analyse it as the data becomes available, and then can feed back to Newcastle about how to change the experiment. So I said near real time, and that in a sense is how short a package you're going to be uploading onto Carmen. But if you rather than waiting and putting a whole large file, you chunk it, then you can be analysing it at the same time as somebody's running an experiment.
So is it already running on a large cluster?
It's currently running on two servers, which so there's 40 nodes on each cluster, and there's one in York and one in Newcastle. Now, the design of it is such that we were intending in the future that it would become federated. So in fact, the Carmen would sit over the top of servers that may sit in other places. That has the big advantage that if there's a center that's generating very large amounts of data, they may not want to keep on transferring that to a server somewhere else. So the easiest thing is to actually sit Carmen over the top of your own server, and the data then can become exposed to the whole Carmen community. Because we use Storage Resource Broker, in fact, it doesn't really matter where the data are.
Okay.
Sorry, was that your question?
I think we'll have to finish it out. I'm sure there are many other questions. I actually had one which I will sacrifice, as you will have to, hopefully. But on the boat trip, maybe there's possibilities to ask Colin. So the questions we have. So thanks very much, Colin.
Okay.